Hello, my name is Jayra Penaloza. I'm the Creative Inquiry Program at Clemson University. I've been a member of the Bios of the Lab since 2022, where I study mitochondrial genomics. Today I'll be showcasing my work of the past 2 semesters, where I annotate and analyze the complete mitochondrial genome of the leopard shark, Trichus semifasciata. I will start by introducing some background information on both mitochondrial genomics and of Trichus semifasciata, followed by a brief explanation of the methods used, including a list of bioinformatics programs. I then proceed to share my findings and with a brief explanation of the importance of this work. So as you can see in the image to the right, mitochondrial DNA is composed of small, circular, double-stranded DNA molecules. Furthermore, mitochondrial genomes are advantageous in research as they're easy to isolate and can be extracted using non-invasive methods, which are important characteristics when working with endangered or rare and difficult to capture species. Additionally, since mitochondrial genomes have multiple copies per cell, fast mutation rate, and are inherited exclusively from the maternal line, the process of tracing genetic lineages among related species is simplified. Thus, using mitochondrial markers, we can infer gene field populations, data that is most relevant to guide conservation efforts in endangered species, for example. Trichus semifasciata is found in the eastern Pacific Ocean, ranging from Oregon in the United States to Mazatlan in Mexico, where they commonly occupy a basin estuaries. Leopard sharks opportunistically feed on benthic invertebrates and small fish. They are characterized as strong active swimmers that form large and nomadic schools of elasmobranchs, ranks, including bat rays, Melobastis californica, and smoothhound sharks, Mustallus marcellus. Despite being active swimmers, this species exhibits limited long-distance movement that results in low genetic exchange among distant populations, for example, between Humboldt Bay and Elkhorn's Low in Northern California. Unfortunately, the leopard shark is threatened by anthropogenic activities. There are currently 38 wastewater treatment facilities that discharge wastewater effluent into the San Francisco estuary, which is a critical nursing site for female leopard sharks that already rely on basin estuaries for reproduction. Wastewater effluent alters water composition and nutrient availability. Changes have caused a decline in leopard shark science in the San Francisco estuary. Furthermore, in recent years, this wastewater effluent has been charged as a culprit of algal blooms in the San Francisco estuary, contributing to die-off of marine species. As you can see, the image at the top shows what an algal bloom looks like from a bird's eye view. So previous to the algal bloom, the water, which was more of a crystalline green color, but has now transitioned to this opaque, murky, brown, rusty color. And unfortunately, what algal blooms do is that they make images like the one depicted here on the bottom become a common scene, where now along the shorelines in the San Francisco estuary, you have an increased number of dead species that unfortunately arrive on shore. Unfortunately, leopard shark is also threatened by overfishing, as pups are poached for the private aquarium trade, as they're coveted by collectors for the distinctive markings. Furthermore, leopard sharks commonly become entangled in the gillnets and longlines of commercial fisheries. So this image to the right is of a thrasher shark that became unfortunately entangled in a gillnet. And gillnets are hung vertically so that the fish get trapped in it by their gills, because they'll swim right in it, and as you can see, they're stuck via their gills. So despite all this, U.S. population is considered to be stable after the enactment of protective laws in California during the 1990s, which include minimum size and daily bag limits, the criminalization of private leopard shark collection, and the prohibition of gillnets along the California coast in waters shallower than 110 meters. Despite all this, there's still growing concerns for the species due to low genetic connectivity among distant populations, since increased genetic connectivity can buffer local populations against the loss of genetic diversity via genetic drift for species experiencing population declines. Additionally, few genetic resources exist for Trichus semifasciata and the Trichidae family as a whole. So our study had 4 objectives. We wanted to assemble, annotate, analyze, and report. So to assemble, we retrieved raw sequence datasets of the epidermal microbiome of Trichus semifasciata, generated by Dona et al. 2022 from GenBank to then assemble the species mitogenome. We retrieved a total of 1,713,261 PE reads from GenBank, and then using this pipeline get organelle, assembled and circularized the mitochondrial genome of the leopard shark with a coverage equal to 34x. To annotate, we wanted to identify the number, type of genes present, and their location in the mitogenome. To analyze, we want to identify and differentiate between conserved and unique mitochondrial features. Lastly, we want to report, which means that we want to create a research for conservation biology. So this slide's an overview of the different bioinformatics programs used. Mitops 2 provides an annotation of the entire genome that includes the location of protein-coding genes, ribosomal RNA, transfer RNA, and the origin of replication for both heavy and light strands. MegaX is a tool that, among other things, visualizes nucleotide sequences and can calculate nucleotide usage or the percentage of each nucleotide present in mitogenome. The genome is then visualized using the program GenomeVX. The codon usage sequence manipulation suite and easy codon web servers were used to determine codon usage of the protein coding genes. The software KK was calculated was then used to conduct analyses of selective pressures on the protein-coding genes. Forna, the software MITFE implemented in Mitos 2 predicted secondary structures of the transport RNAs that were visualized with Forna. Tanner repeats finder and microsatellite repeats finder were used to identify the presence of Tanner repeats and microsatellites respectively in the control region. Lastly, the RNA fold web server predicted the secondary structures in the control region. So as the standard in studies describing mitochondrial genomes, a visual map, as you can see depicted here on the right, is used to show location, direction, and the type of genes discovered in the mitochondrial genome of tracking fatty acid. So in purple we have the ribosomal RNAs, in pink we have the transport RNAs, and in green we have the protein-coding genes. So the newly assembled mitochondrial genome of Leopard shark is 16,613 base pairs long and contains the expected composition of 22 transport RNAs, 2 ribosomal RNAs, and 13 protein-coding genes. As expected, most of the protein-coding genes were situated on the heavy strand. Nucleotide composition in mitogenome was determined to be AT-rich, with the AT content equal to 61.1%. AT contents of 60% have been reported before in the cofamilial species Mystelis minusa or the star-spotted smoothhound shark. Our next step is to determine codon usage in the protein-coding genes. The most frequently used codons include ATT, which codes for isoleucine, ATA that codes for methionine, and CTA, CTT, and TTA, which all code for leucine. The least frequently used codons include CCG, which codes for proline, TCG that codes for serine, and AAG, which codes for lysine. The codons in the protein-coding genes of Trichocephala fasciata were observed to be AT-rich. These findings are in line with previous observations in the cofamilial Hematricus japanica, or the Japanese toad shark, and Cephalosalem umbertili, or the Japanese swell shark. RSU stands for Relative Synonymous Codon Usage, and essentially what this does is it evaluates synonymous codon bias. And this figure here to the right is a figure depicting RSU values. And RSU is a value where if it's greater than 1, then it could indicate that a codon is potentially being overrepresented in the mitochondrial genome. But if it's less than 1, then it can indicate that this codon is potentially being underrepresented in this mitochondrial genome. Selective pressures on each protein-coding gene were then analyzed using the program KKS calculated, which provides us with 3 values. It gives us Ka, Ks, and ratio Ka, Ks, also known as omega. Ka is the number of non-synonymous mutations per non-synonymous site, whereas Ks is the number of synonymous mutations per synonymous site. If omega is less than 1, protein-coding genes are predicted to be under negative or purifying selection, which means that amino acid substitutions that improve protein function are being eliminated via natural selection. However, if omega is greater than 1, protein-coding genes are predicted to be under positive or diversifying selection, which means that natural selection is favoring amino acid changes that could potentially improve fitness or function of the gene. So this figure to the right depicts what a synonymous substitution and what a non-synonymous substitution are. A synonymous substitution is one where, despite the fact that you're changing one of the nucleotides in the codon, you're still coding for the same amino acid, so you're not necessarily altering protein function. Whereas non-synonymous substitution is one where you're now changing one of the nucleotides, but now you're coding for a different amino acid, which is going to alter protein function. Another case of a non-synonymous substitution is one where you're originally coding for an amino acid, but now you're switching one of the nucleotides such that you now have a stop codon, which results in an incomplete protein. So this figure here depicts the Ka-Ks ratios for 13 protein-coding genes in Trichocephala fasciata. All of the Ka-Ks ratios do not surpass 1, indicating that all the protein-coding genes are experiencing purifying selection. However, NAD3 and ADD6 automatically stick out because their values are comparatively larger when you look at the remaining 11 protein-coding genes, as their values are the ones closest to 1. What this means is that in NAD3 and ADD6, purifying selection is the weakest. In a similar manner, COX-1 sticks out because its value is so much smaller than the remaining 12 protein-coding genes, and its value is the one closest to 0. What this means is that purified selection is the strongest in this gene. Selective pressure analyses have rarely been conducted in sharks, but 2 recent studies in the Japanese swell sharks, Cephalosalem umbertili, and the Chilean devil-worm Mobila terraplicana found purifying selection for almost all protein-coding genes. So this slide depicts the secondary structure of tRNA molecules. So you have an acceptor stem, a T stem, an anticodon stem, and a D stem, and every stem, with the exception of the acceptor stem, has a corresponding loop. So you have a T loop, an anticodon loop, and a D loop. And in place between the acceptor stem and T stem, you can have a variable loop. This variable loop can be large enough to form an additional helix, or it can be very small, as depicted here. And so here to the right, I have a picture of what a truncated tRNA and what a normal tRNA would look like. So a truncated tRNA, as you can see, it's missing the D stem and in place has a simple D loop. The secondary structure of tRNAs exhibit a typical cloverleaf structure, with the exception of tRNAS1, which was observed to have a missing D arm. So Phenylcylium umbretellii, a member of the same order as Trichostomia fasciata, also exhibits truncation of the tRNAS1 gene. Truncation of tRNAS1 is not unexpected, as the D-loop arm and entire stem of the tRNAS1 gene is often missing in metazoans, including sharks and rice. A putative control region located between TRNP and TRNF in the mitochondrial genome Trichostomia fasciata is 974 base pairs in length. Leopard shark control region exhibits a neural base composition where T is equal to 34.5%, C equals 20%, A is equal to 31.5%, and G is equal to 14%. Its high A plus T content has often been reported in vertebrates. Furthermore, the control region is repetitive with a 46 base pair long tandem repeat and 16 microsatellites composed of AA and TT dinucleotide repeats. Additionally, the RNA fold web server predicted 20 possible secondary structures in the control region that contain stem, loop, or hairpin structures. However, there's very few studies characterizing the control region of sharks. Therefore, we argue in favor of additional studies in order for us to better be able to understand the role of the control region in general replication and transcription. And this figure to the right is just one of the predictions of the control region's secondary structure. So mitochondrial genomes are useful population genetic studies in endangered species. Comparative studies of mitochondrial DNA can identify population changes and help scientists develop short-term and long-term conservation plans. To determine the genetic variation in mitochondrial genome in different populations, researchers can determine migration patterns and the flow of genetic information among populations, which are essential for managing populations and threatened species. This newly assembled mitochondrial genome of the leopard shark can be used as a reference to bioprospecting and biomonitoring of Trichostomia fasciata, using indirect surveillance methods such as environmental DNA. Environmental DNA is really neat because since we're specifically working with an aquatic species, what environmental DNA allows us to do is it allows us to take water samples, and from those water samples to then retrieve the genetic code for tens or even hundreds of different species. And this is because species will naturally release feces, urine, and shed their skin into the water column. And these DNA analysis is really neat because it allows us to conduct biodiversity surveys, can allow us to detect new species, and to conduct bioassessment and biotic indices, all while not using any direct sampling strategies, which allows us to reduce the stress that we induce on species that are already facing anthropogenic activities. This study for the first time assembled, characterized, and analyzed the complete mitochondrial genome of the leopard shark, which will serve as a genomic resource to support conservation efforts focusing on Trichostomia fasciata. This study represents a step forward towards implementation of direct surveillance in the leopard shark. I would like to thank Clemson Creative Inquiry, the Honors College, the Department of Biological Sciences, and Dr. Baeza for their guidance and support of this project. Thank you for watching.